All right, well, welcome to lesson 21.2, Protist Structure and Function. We're going to be discussing the characteristics of the domains and kingdoms of living organisms. That's our overall goal. And take a look at the far side. It's kind of humorous. All right, so to begin with, our objectives are to describe the various methods of protist locomotion and also describe how protists reproduce. First, let's think about it. Protists move, sense the environment, digest food, and even reproduce, all within confines of a single cell. Imagine what each such cells would have to be like to succeed in the never-ending struggle for life on Earth. And protists are the winners in that struggle. So our first objective is to describe the various methods of protist locomotion. Many unicellular protists move by changing their shape, a process that makes use of cytoplasmic projections known as pseudopods. The cytoplasm of the amoeba, for example, streams into this pseudopod and the rest of the cell then follows. This type of locomotion is called amoeboid movement and is found in many protists. Amoeboid movement is powered by a cytoskeleton protein called actin. Actin also plays a role in the muscle contractions of animals. Many protists move by means of cilia and flagella, structures supported by the microtubules. Cilia are short and numerous, and they move somewhat like oars on a boat. Flagella are relatively long and usually number only 1 or 2 per cell. Some flagella spin like tiny propellers, but most produce a wave-like motion from base to tip. Protists that move using cilia are known as ciliates, and those that move with flagella are called flagellates. Some protists are non-motile. They depend on air or water currents and other organisms to carry them around. These protists form reproductive cells called spores that can enter the cells of other organisms and live as parasites. Spore-forming protists include Plasmodium, which is carried by mosquitoes and causes malaria. This is what Plasmodium looks like. And Cryptosporidium, over here, which spreads through contaminated drinking water and causes severe intestinal disease. Our first objective, again, was to describe the various methods of protist locomotion. Some protists move by changing their cell shape, and some move by means of specialized organelles. Other protists do not move actively, but are carried by wind, water, and other organisms. Objective number 2 is describe how protists reproduce. All right, so amoebas and many other protists reproduce by mitosis. They can just duplicate their genetic material and then divide into genetically identical daughter cells. Mitosis enables protists to reproduce rapidly, especially under ideal conditions. But it produces cells that are genetically identical to the parent cell and thus limits the development of genetic diversity. Paramecium and most ciliates reproduce asexually by mitotic cell division. However, under stress, paramecium can remake themselves through conjugation, sort of like bacteria can do, a process in which 2 organisms exchange genetic material. After conjugation, the cells then reproduce by mitosis. Paramecium has 2 types of nuclei, a macronucleus and 1 or more smaller micronuclei. The micronucleus holds a reserve copy of every gene in the cell, sort of like the plasmid has in the bacteria. A macronucleus has multiple copies of the genes the cell uses in its day-to-day activities. So what happens here? The steps are listed, so we're just going to go through them one at a time. Number 1, conjugation begins when 2 paramecium are attached to each other. Number 2, meiosis of their diploid macronuclei produces 4 haploid macronuclei. Okay? 4 haploids. Number 3, in each cell, 3 of the haploid micronuclei disintegrate. They're not used. The remaining micronucleus in each cell divides then by mitosis. This then divides. The 2 cells exchange 1 haploid micronucleus from each pair. In each cell, the micronuclei fuse to form a single diploid micronucleus, and the macronuclei then disintegrates. These fuse together and that disintegrates. Each cell forms a new macronucleus from its micronucleus then. And that is what you have for conjugation. Conjugation is not a type of reproduction because no new individuals are formed. Conjugation is, however, a sexual process because new combinations of genetic information are produced. In a large population, conjugation helps produce and maintain genetic diversity. Many protists have more complex sex life cycles in which they alternate between a diploid and a haploid phase in a process known as alternations of generations. A water mold is an example of a protist that undergoes alternations of generations. Water molds grow into long branching filaments consisting of many cells formed by mitotic cell division. They reproduce asexually by producing spores in a structure called a sporangium, right here. In water molds, the spores are flagellated, which means they have little tails on them. Water molds also reproduce sexually by undergoing meiosis and forming male and female structures. The male and female structures produce haploid nuclei that fuse during fertilization. Oh, went too far, didn't I? That then fuse during fertilization, forming a zygote that then begins a new life cycle. So our objective was to describe how protists reproduce. Well, some protists reproduce asexually by mitosis. Others have life cycles that combine asexual and sexual reforms of reproduction. All right, ladies and gentlemen, I think I forgot to put the objectives at the end. No, here they are. So I hope you know these two objectives: describe the various methods of protist locomotion and describe how protists reproduce. And our goal is to discuss distinguishing characteristics of domains and kingdoms of living organisms. And this time we are focusing, obviously, on the protist. All right, ladies and gentlemen, have a good evening.